Hello friends, today we will discuss about enzymatic nature of RNA, that is ribozyme. Then we will discuss about deoxy-RNA. Then we will discuss about nucleotide, polynucleotide, then nucleoside, dinucleotide, and then we will discuss about purine, pyrimidine, and lastly we will discuss about different types of bone that are formed within a dinucleotide. So let's start. RNA is a type of nucleic acid that shows the behavior of an enzyme, that shows enzymatic behavior. And due to enzymatic nature of RNA, it is also known as ribozyme. And this ribozyme catalyzes the specific biochemical reaction like RNA splicing, translation. But this deoxy-RNA does not behave like an enzyme because it is evolved from RNA through chemical modification, and this chemical modification occurs in the strand of RNA. Therefore, single RNA strand converted to double-stranded RNA, and then chemical modification occurred in the nitrogenous base of RNA. Therefore, uracil replaced by thymine, and lastly, the chemical modification occurred in the ribose sugar of RNA. Therefore, the degeneration of oxygen atom from second carbon of ribose sugar, which leads to the final structure, this one is ribose sugar, this one deoxyribose sugar. That means degeneration of oxygen atom from the second carbon. Okay, now DNA. Similarly, we will discuss about DNA. D means deoxyribo and A means nucleic acid. DNA is a special and advanced type of nucleic acid because of all this modification. So the nucleic acid is actually the polymer of nucleotide. Yeah, polynucleotide, that means. And this nucleotide has 3 units. One is deoxyribose sugar, and second one is adenine, that is the nitrogenous base, and third one is phosphate molecule. And this glycosidic bond, it connects the deoxyribose sugar with adenine and form the structure called nucleoside. And this glycosidic bond is formed between carbon and nitrogen. Therefore, this bond also known as covalent bond. Similarly, the ester bond, that is formed between the nucleoside and phosphate molecule. This ester bond connects the nucleoside with phosphate molecule. Now, this phosphodiester bond, it connects the 2 nucleotides. Suppose this whole structure is dinucleotide. This one nucleotide, suppose B, this one nucleotide A. Dinucleotide means nucleotide. Here is the nucleotide A, this one nucleotide A, and this one nucleotide B. So the 5' to 3' phosphodiester bond, it connects the nucleotide A with nucleotide B to form a polynucleotide. And this phosphodiester bond is formed between the carbon and oxygen molecule. Therefore, this bond is a covalent bond, and covalent bond always formed between non-metals. Now, we will discuss about purine and pyrimidine. Actually, the hydrogen bond that is present in between the purine and pyrimidine, this hydrogen bonds, it connects the purine of nucleotide, say A. This hydrogen bond connects the purine of nucleotide A with pyrimidine of nucleotide B. This hydrogen bonds connects the purine of a nucleotide with pyrimidine of different nucleotide to provide stability to the region. That means this hydrogen bond is providing more stability. Now finally we will discuss about the total possible bonds that can be formed within the dinucleotide. So here is the hydrogen bonds. This one, the hydrogen bonds in dinucleotide. Total possible hydrogen bond can be 5. Suppose this one purine, suppose A adenine double bonds with thymine or uracil in case of RNA. So total hydrogen bond will be 5, and these hydrogen bonds connect the purine and pyrimidine. That means this hydrogen bond connecting the two nucleotide chain. Suppose here is the nucleotide chain 1, then chain 2. Hydrogen bond will connect these two nucleotides. Suppose this one nucleotide A, this one nucleotide B. So this hydrogen bond will connect this two nucleotide chain to provide more stability. So in case of dinucleotide, there would be total 5 hydrogen bond. And ester bond, this is the ester bond that is formed between carbon and oxygen. Ester bond will be 2. Suppose this is a nucleotide A, here is the one ester bond. Similarly, one ester bond will be here. So therefore total 2 ester bond, and this ester bond connects the phosphate group with this deoxyribose sugar. This ester bond connects the phosphate group and deoxyribose sugar. But actually this phosphodiester bond is a type of ester bond only because in this dinucleotide structure, this for 5 days to 3 days phosphodiester bond, it connects the two nucleotide. Suppose nucleotide A and nucleotide A is connecting with nucleotide B with the help of this bond, phosphodiester bond. And lastly, glycosidic bond. Similarly, glycosidic bond will also be 2, and this glycosidic bond is formed between deoxyribose sugar and adenine. That's all. So please do subscribe the channel for more updates. Thank you.